The acid-fast stain is a differential stain used to identify bacteria that possess high concentrations of mycolic acids in their cell walls, so-called acid-fast bacteria. The waxy nature of these cell walls prevents uptake of many dyes, including those used in the gram stain procedure. The lipid-soluble dye carbolfuchsin is used as the primary stain in the acid-fast procedure. Carbolfuchsin is a red dye that penetrates all cell walls, including those containing mycolic acids. Cells that retain the primary stain after decolorization are called acid-fast because they hold fast or tightly to the primary stain. These acid-fast bacteria stain red and include members of the Nocardia and Mycobacterium genera. Species that do not retain the primary stain after decolorization are called non-acid-fast. These cells are stained blue by the methylene blue counterstain. Two methods are commonly used for acid-fast staining. The Ziehl-Neelsen method uses heat to drive carbolfuchsin into the cell cytoplasm. The Kinyoun, or cold method, uses the chemical turgitol instead of heat. Here, we'll use the Ziehl-Neelsen method. Prepare a smear of your culture. Let the slide completely air dry and then fix it. The Ziehl-Neelsen method involves heating carbolfuchsin. Because carbolfuchsin contains phenol, you should do this heating step in a fume hood if one is available. Use a hot plate to heat a beaker of water. Place a stain rack across the top of the beaker. Once the water is boiling, use a clothespin to lay your slide, smear side up, across the stain rack. Place a piece of filter paper over the smear and saturate the paper with carbolfuchsin. Reapply carbolfuchsin as it evaporates. The filter paper must stay wet throughout the procedure. Typically, the smear is heated with carbolfuchsin for 5 minutes. Your instructor will provide details on the procedure used in your lab. Carefully remove the filter paper with forceps and place it in a designated waste container. Your instructor will advise you of the disposal procedure used in your lab. Let's look at what happens to the cell walls at the molecular level when the smear is heated with the primary stain carbolfuchsin. Carbolfuchsin is a mixture of phenol, which is also known as carbolic acid, and an assortment of red chemicals called fuchsin. Heat softens the waxy mycolic acids of an acid-fast cell wall and helps fuchsin to penetrate and color acid-fast bacteria. Fuchsin also penetrates and colors non-acid-fast bacteria. At this step in the protocol, both types of bacteria stain red. Allow the slide to cool. Rinse the slide with distilled water to remove excess stain. Tilt the slide and aim the water above the smear. Let the water wash over the smear. Decolorize the smear with acid alcohol by dropping the decolorizer onto the smear until no more stain runs from the smear. Let's look at what happens at the molecular level when acid alcohol is added to the smear. Acid alcohol is an intense decolorizing agent that penetrates the peptidoglycan layer of bacterial cells and washes out stain from the cell cytoplasm. The mycolic acids and other lipids of the acid-fast cell wall, however, impede the entry of acid alcohol. After decolorization, the acid-fast cell remains red. In the non-acid-fast cell wall, there are no mycolic acids to impede the acid alcohol from penetrating the peptidoglycan and plasma membrane of the cell. Acid alcohol is able to wash out the carbolfuchsin primary stain from the cell cytoplasm. After decolorization, acid-fast cells retain the carbolfuchsin and remain red. Non-acid-fast cells lose the dye and become colorless. Rinse the slide briefly to remove the decolorizing agent by letting distilled water wash over the slide. Cover the smear with methylene blue counterstain for 1 minute. Staining times may vary. Your instructor will provide details on the procedures used in your lab. Methylene blue is used as the counterstain in the acid-fast staining procedure. Let's look at what happens at the molecular level when methylene blue is added to the smear. Methylene blue is a water-soluble dye that cannot penetrate the waxy mycolic acid layer of acid-fast cells. Thus, at the end of the acid-fast stain, acid-fast cells remain red from the carbolfuchsin primary stain. This is not the case with non-acid-fast cells. Methylene blue easily penetrates the cell wall of these cells, staining them blue. Red acid-fast cells can now be distinguished from blue non-acid-fast cells. Briefly rinse the slide with distilled water and gently blot the slide dry by placing it between bibulous paper. Do not wipe the slide, just blot. You can now examine your stained bacterial smear under the microscope.